Hello students, welcome to CBSE NCERT Solutions by Vedantu. This is Baswaratso, your biology master teacher. Now students, in the previous class, we did cell cycle and cell division first part, right? All of us did the first part there, where we did about something called as the entire cell cycle, and also we finished off something called as the mitosis, right? So the mitosis has been now been over. Let me just check students. Let me know if I'm audible and visible to all of you, first of all. Let me know if I'm audible and visible to all of you. If I'm audible and visible. Yes, I'm audible, students. Sound check, sound check. Audible, I don't think so. Okay, let me check again. Let's start streaming here. I don't know why it's not streaming. Okay, let me just check one more time. Sound check. Okay, it's working perfectly. Now, students, in the first part, we finished off with something called as the entire mitosis was over with the DNA content, and all of you saw the DNA content and everything. You are like, okay, fine, sir. It makes sense. What is we call why we call it as a equational division. Equational division. Now, in today's class, we'll be learning something called as reduction division. Right? What exactly is this reduction division, and where does it happen? When does it happen? What are the different stages in reduction division? That is meiosis. Because all of you know that when it comes to meiosis, right, there is something called as prophase one, and this Prophase I has been a nightmare for so many students. I repeat again, it has been a nightmare for so many students that they still get confused between Prophase I. As in, like, what are the different steps in Prophase I? How do we proceed with what is leptotene, what is zygotine, what is pacatine, what is diplotene, what is diakinesis? Students, in this today's class, all that confusion is going to get clear. That you will know exactly what is leptotene, what is zygotine, what is pacatine, and with the help of NCERT lines. Okay, so let me know in the chat if you are ready for today's class. Ready, all of you? If I'm properly audible? Yes. I have to check my sound more time. You're perfectly. Yes. I'm perfectly audible and visible to all of you. Okay. Now, students, let's read what lines of NCERT. Okay. Now, let's go to red option. Yes. Now, meiosis. Right, meiosis. The production of offspring by sexual reproduction includes the fusion of two gametes. Fusion of two gametes. That is, the male gamete, that is the sperm, right? The male gamete, which is the sperm, and the female gamete, which is the egg, right? The male gamete, which is the sperm, and the female gamete, which is the egg, which end up fusing and produce the a first cell, that is the zygote. They end up producing the zygote. Now this zygote eventually develops and becomes a multicellular structure. Becomes a multicellular organism. That is how it goes. That's how it goes, right? Now what is given here? 23 chromosomes are present here only. Nothing more, nothing less. Okay. Now are formed from the specialized diploid cell. Okay. Now this kind of a cell, this kind of cell division that reduces the chromosome number. That is, from diploid we're getting haploid, right? From diploid we're getting a haploid set of cells, diploid to haploid. That chromosome number is half as a result of production of haploid daughter cells. This is called as meiosis. So what exactly happens in most students? In the case of meiosis, there is a change in the number of chromosome. From the diploid number of chromosomes, we are getting haploid number of chromosomes. Diploid to haploid number of chromosomes. Okay. Now, meiosis ensures the production of haploid phase in the life cycle of the sexually reproducing organisms. Very important line, because imagine students, see, the sperm and the egg are produced by a diploid organism, that is humans. Humans were diploid. From diploid we have haploid again. If this conversion did not happen, the zygote would develop into something called as 4n. It would become 4n, right? So imagine there was no change in ploidy. Even the gametes were also 2n. Then the zygote would end up becoming 4n. Zygote would end up becoming 4n. To encounter this, to make sure the zygote is not becoming tetraploid or something, right? There is something called as meiosis, and the gametes are haploid nn. So we end up getting zygote right now is 2n. So the haploid phase in humans is the gametes. Okay. Whereas fertilization restores the diploid phase. Obviously, fertilization is making sure the zygote is going to be diploid. So humans are diploid, zygote is diploid. The only haploid phase is going to be the gametic phase. It's going to be the gametic phase. Clear? Now we come across meiosis during the gametogenesis in plants and animals. All of you know spermatogenesis, oogenesis, right? This lead to the formation of haploid gametes. The key feature of meiosis are follows. What are the key features of meiosis? Understand. Now, meiosis involves two subsequent sequential cycles of nuclear and cell division called as meiosis 1 and meiosis 2. Students, understand, if you want to broadly classify meiosis, if you want to broadly classify meiosis, it can be divided into two categories. Two categories. The first one is called as interphase. The first one is called as what? Interphase. And the second one is called as meiosis. Meiosis. This meiosis again is further divided into two categories. Into two categories. That is basically the meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2. Now all of you know this interphase has G1, S and G2. All are present in interphase. Now between meiosis 1 and meiosis 2, between meiosis 1 and meiosis 2, there is one more phase here. The phase between meiosis 1 and meiosis 2 is called as, anyone in the chat who can tell me? Anyone? That is basically interkinase. Interkinesis. Interkinesis is between the meiosis 1 and meiosis 2. Now if you remember, tell me students, in the case of interphase, in which phase of interphase the DNA duplication happens? Can anyone remind me quickly? In which phase? Tell me, in which phase of interphase DNA duplication happens? All of you remember? In the S phase, right here. In the S phase, there is DNA duplication. DNA duplication happening here. Absolutely. Now, since interkinesis is also between meiosis 1 and meiosis 2, do you think DNA duplication happens in interkinase? No. So remember students, interkinesis is just like interphase. Interkinesis is just like interphase, but in the case of interkinesis, there is no DNA duplication. No DNA duplication. No DNA duplication in the case of interkinase. I hope that point is clear to all of you. The main difference between interphase and interkinase. Interkinase DNA duplication is happening. Interphase, it is interkinase, it is not happening. Interphase, it is happening. Here it is happening. DNA duplication. Now let's see what is given in NCERT, all of you. Let's see what is given in NCERT now. Okay. Meiosis involves two sequential cycles of nuclear and cell division called as meiosis 1 and meiosis 2. All of you know. Meiosis 1 is initiated after the parental chromosomes have replicated. That is, meiosis 1 is happening after the S phase and G2 phase. So after the DNA duplication has happened, then only meiosis 1 will happen. So meiosis 1 is initiated after the parental chromosomes are replicated. Where? In the S phase. In the S phase. Now meiosis involves pairing of homologous chromosomes, which we will see in the case of your zygotine. Right? Between then we'll also see crossing over in pachytene, right? And final product. The final product of meiosis is 4 haploid cells. 4 haploid cells are the final product here. Any doubt till now? Tell me, students. Any doubt till now? So Joe level is low because I'm sitting, because the mic is not there today, right? Usually you can hear my voice very clearly because mic is there. Now today I'm in my house. Today I'm sitting in my house, right? I'm seeing the comments. Don't worry. Okay. So since I'm sitting in my house, there is no mic here. That is why the voice is not very. It is not going directly to your ear. The voice is being diffused. Right. Little bit of physics here and there. Okay. Now all of you know. We learned there is meiosis 1 and meiosis 2. Okay. Meiosis 1 and meiosis 2. Now meiosis 1 and meiosis 2 has multiple parts. Like how in the case of mitosis we had PMAT. Remember, In the case of mitosis, we had something called as PMAT. All of you remember, I believe. In the case of mitosis, we had the PMAT, right? That is prophase, prophase, metaphase, anaphase, and telophase. Did we have this? Yes. We had the following phases when we studied about the in the mitosis. Similarly in the case of meiosis also, we have the following. We have in meiosis 1, under meiosis 1 we have the prophase 1, metaphase 1, anaphase 1 and telophase 1. Similarly in meiosis 2 also we have something called as prophase 2, metaphase 2, anaphase 2 and telophase 2. Clear? Now we will understand. Now we will understand what exactly is happening in meiosis 1. Students, I want everyone to be very careful now. Let me tell you, whatever lines you learn right now, yeah, all of these lines, right? Questions will come directly from here. I'm not even kidding. Questions will come directly from here, and you will be like, okay, fine, it is such an easy question. It will come direct, it will come. So please pay extra attention right now when we studied Prophase 1. Now, understand. Students, this is a very small trick. They can ask you direct question also. Your prophase 1 can be further subdivided. Prophase 1 can be subdivided into different types of, again, levels. Okay. For example, I'll tell you a small trick. Let zebra play during the day. During the day. Now here the L stands for leptotene. Leptotene. The Z stands for zygotine. Zygotine. The P stands for pacatine. Pacatine. The D stands for diplotene. Diplotene. And final D stands for diakinesis. Diakinesis. Now students, this order is also very important. In examination, sometimes they can ask directly the order also. They can ask you tell me the different order of prophase 1. So you're supposed to tell the proper order. Leptotene, zygotine, pacotine, diplotene and diakinesis. The same order has to be maintained. And what is the trick, students? Let zebra play during the day. Let zebra play during the day. That is a small trick. And we can go in a similar order. Similar order. Okay. Now understand what have happened. Any doubt till here? Quickly ask me. Tanuja, Krishna, any doubt till here? Any doubt till here? Quickly ask me. No doubt. Yes. Krishna. Krishna is such a nice name. Krishna is such a nice name. Okay. Any doubt till here, students? Quickly ask me. Any doubt till here? Okay. Now let's read what is happening in leptotene now. Now during leptotene, I'll tell you the exact line of NCERT where question will come from. Okay. I'll tell you the exact line of NCERT where the question will come from. Okay. During leptotene, the first stage leptotene, the chromosomes become gradually visible under the light microscope. This line right here is a PYQ. Previous year question right here. They'll ask you, in leptotene, chromosomes become gradually visible in light microscope. Assertion. Assertion, that's all. That is a simple assertion. The reason they can use something else. But they'll ask you, is assertion correct? Understood? So in leptotene, students, what is happening? Chromosomes are becoming gradually visible under light microscope. Chromosome becoming gradually visible under light microscope. Now, the compaction of chromosome continues through leptotene. So, what is happening, students? Chromosomes are becoming visible and there is condensation of chromosome. Compaction of chromosome. Clear, all of you? Is leptotene clear? Two main points. Two main points. First point is what, students? The first point is chromosomes are becoming gradually visible. The second point is that the compaction of chromosome is happening. Compaction of chromosome is happening. Only 2 points of leptotene and that's all. And that's all. Now students, before we see, This is your chromosomes. Chromosomes are becoming gradually visible under light microscope. This is leptotin. Okay. Now, understand zygotin with the help of a small diagram. Now, students, in your class, whenever there is an activity, whenever there is an activity in your class, they will ask you to make a group of two. Tell me, what is that? Roshni, you tell me. In your class, has they ever played a game and where they ask you to make a group of two? They'll ask, make a pair with someone. That time you will go and find your best friend. You'll find your best friend because both of you have some similarities and everything. You'll find your best friend. Okay. So similarly, also in the case of chromosomes, they are trying to find a best friend. That is, two chromosomes are coming together, which extremely look alike. They're like twins. They look like twins. So when the two chromosomes are coming together, the process of two chromosomes coming together, that is called as synapsis. That is called as synapsis, students. Synapsis. Understand? So if we have one chromosome here, we have one more chromosome here. The two chromosomes are coming together. The two chromosomes are coming together. They look alike. They are same to same. When two chromosomes look alike, we call them homologous chromosomes. We call them homologous chromosomes. Homologous chromosomes. Okay. Now here, students, this is a chromatid. This right here is a chromatid. Is a chromatid. These two are called as what? These two are called as sister chromatids. Sister chromatids. Sister chromatids. Now, when the two homologous chromosomes are coming together, the process of two homologous coming together is called as what? Synapsis. So this right here is called as what? Synapsis, students. Synapsis. 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 Okay. Now, students, when the two homologous are coming together, between the two chromosomes, there is some form of complex formation. Can you see? Between the two chromosomes, there is some form of complex formation between two chromosomes. It's like one glue. You take two chromosomes, you can add one glue in the middle. Very quick you add and stick the two chromosomes. This complex right here, can you see this complex right here, green color one? This complex is called as what? It's called as synaptonemal complex. Synaptonemal complex. So understand the difference, students. Understand the difference. Two homologous chromosomes coming together, the process is called as what? Synapsis. The process is called as synapsis. Now between them there is a glue that is or complex that is called as synaptonemal complex. Any doubt here? Quickly ask me, students. So, see, chromatin is the thread-like structure. When the chromosomes are in a interface, right, they appear as threads, beads on a thread. That is called as chromatin. That is called as a chromatin thread. Now, what is that is called as chromatin. Chromatin. Now, can you see this individual one? This is called as chromatid. This individual called as chromatid. This entire thing is called as chromosome. Understood? Chromosome is the entire thing. Individual one line, can you see? This is called as chromatid. Chromatid. What is chromatin? Tin. Tin. Chromatin is the thread-like DNA. Thread-like DNA. Now, what is the? Then I'll ask you one more one. What is the difference between centromere, telomere, centrosome, and centriole? Let's see how many of you can tell. Let's see how many of you can tell. Tell me the difference between centromere, centrosome, centriole, and telomere. Anyone can tell me in the chat. Quickly, how many of you know? Simple question. Simple question. What is what is telomere? What is centromere? What is gonna be your centrosome? What is centriole? And what is all of them? Let's see how many of you. Quickly tell me in the chat. If you do not know, I will tell it towards the end. Very easy it is. Very easy. I will show you with the help of diagram. You will never forget. You don't have to buy heart all this. Okay. Now, students, let's read what is given in NCERT. Okay. Now, the second stage is called as the zygote. All of you know the second stage is called as what? The second stage is called as the zygote stage. Right? The second stage is called as what? Zygote stage. Zygote. During this stage, chromosomes starts pairing together. I told you the two favorite, all of you like how you make teams, right? How you make teams. Here also, the two chromosomes are coming together. That pairing up. The pairing up is called as what, students? So during this stage, chromosomes start pairing together. The process is called as what? Synapses. Synapses, right here. Synapses process. Such paired chromosomes are called as homologous chromosomes, which look alike, same to same, twins they are. Now, the electron micrograph of this stage indicates that chromosome synapsis is accompanied by formation of a complex called as what? What are the complex here, students? Synaptonemal complex. Very quick, very quick. Okay. Now, this complex formed by a pair of synaptic homologous chromosome is called as bivalent. Students, understand the difference again. Don't by heart this. This stage right here is called as what? This stage right here is called as bivalent, or we can also call it as tetrad. We can call it tetrad. Now why? Why are we calling it bivalent first of all? The reason we are calling it as bivalent, the main reason we are calling it bivalent because of it has two chromosomes. It has two chromosomes. That is why we are calling it bivalent. See, chromosome number 1, chromosome number 2. Two chromosomes. Then why are we calling it a tetrad then? The reason we are calling it a tetrad is look at the chromatids now. How many chromatids do we have? We have 1, 2, 3, 4. 4 chromatids. So can I call it tetrad also? 4 chromatids. So I'm calling it tetrad. So this structure right here can be called as bivalent because of 2 chromosomes. It can also be called as tetrad because of 4 chromatids. 4 chromatids. Is that point clear to all of you? Tell me yes or no in the chat, because this is a very important question which has been asked repeatedly in your examination. Repeatedly they'll ask. Don't by heart any of this. Understand. Absorb the concept. Okay. Now, one beautiful question comes now. One beautiful question comes. Tetrad or bivalent stage. Tetrad or bivalent stage is clearly visible in which stage of prophase 1? The question is very simple. Tetrad and bivalent, which stage is it completely clearly visible? Now, if you write zygotine, your answer is wrong. If you write zygotine, answer is wrong. Why? Because remember, students, in zygotine, the formation is happening. In zygotine, formation of bivalent or formation of tetrad is happening. But this bivalent or tetrad, it is clearly visible in the next stage. And the next stage is called as Pacatine. Pacatine. So if I ask you a question, in which stage of prophase 1? In which stage of prophase 1 bivalent or tetrad is clearly visible? What should be your answer, students? What should be your answer? The answer should be Pacatine, sir. Pacatine. Understanding? So bivalent and tetrad formation is happening in zygotine formation, but it is clearly visible in the next stage, that is Pacatine. It's given here, see. However, these are more clearly visible in the next stage. The first two stages of prophase 1 are relatively short duration. The next stage is called as Pacatine. So bivalent tetrad is clearly visible in Pacatine. Beautiful line of NCERT. And remember, students, your leptotine and zygotine, both the stages are very short-lived. Very short-lived. Okay. Now, Pacatine now. What happens in Pacatine? During this stage, the 4 chromatids of each bivalent, 4 chromatids, see this is homologous chromosomes. Homologous chromosomes is pairing, and between them is what? Between them is the synaptonemal complex. Can you see? Gum. Synaptonemal complex is there. Okay. Now, what happens in Pacatine, students? Pacatine is very beautiful. Understand, understand. Understand, I'll teach you, I'll draw and teach you, understand. Okay, Pacatine. We'll use color. I hope the color is visible, okay? Here we have one chromosome. We have one chromosome here, okay? And we have one more chromosome here. It became like Karnataka flag color, nice. Okay. We have two different chromosomes here. They are homologous chromosomes. They are homologous chromosomes. The homologous chromosomes. What are these two, students? Tell me. They are called as a sister chromatid. Sister. Now, what about these two? One from here and one from here. What about these two? This one and this one. They are called as non-sister. They are called as what? Non-sister chromatids. Non-sister chromatids. One from here and one from here. Now, what happens is you're exchanging. When you're bringing, let's say, Tanuja is bringing biryani, okay, and Roshni is bringing a sandwich. Roshni is like, take my sandwich and give me biryani. So they're doing exchange offer. They're doing exchange offer. Now here also something is exchanging. What is exchanging? The chromatid is exchanging. So what is happening here is this chromatid right here. This chromatid right here is going this side, and this chromatid right here is coming this side. There is something called as exchange of chromatid is happening. Exchange of chromatid. Okay, understood? Now before the exchange of chromatid happens, the two chromosomes have to come close to each other, right? So I'll just draw the final thing. I'll draw you students understand. Here we have the, imagine they're come close together and the exchanges happened now. Exchanges happened. Writing on the board is so much easier, I swear. Students, can you see the difference now? Students, can you see the difference here? This particular chromatid has come this side. This chromatid has come this side now. And what is present between them? Between them, we have the synaptonemal complex. We have the synaptonemal complex between them. Now, what exchange? What exactly happened here? There was exchange of genetic material. Exchange of genetic material between non-sister chromatid of homologous chromosome. This entire process is called as what? Crossing over. Simple, very simple. It is called as crossing over. Read what is given here. During this stage, four chromatids of each bivalent of chromosome become distinct and clearly appear as tetrad. I told you, Packeting stage, right? This stage is characterized by appearance of recombination nodules. So, since crossing over is happening here, crossing over is happening here. At the site of crossing over, we have what? At the site of crossing over, we have the recombination nodules at the site of crossing over. Recombination nodules. Recombination nodules at the site of crossing over. Okay. Now, the site at which crossing over occurs between non-sister chromatids. See, non-sister chromatids of homologous chromosome. Crossing over is the exchange of genetic material. It is the exchange of genetic material between what? Between, where is it? It's genetic material between the two homologous chromosomes. See, homologous chromosome number 1, homologous chromosome number 2. Between homologous chromosomes, crossing over is also enzyme mediated process, and the enzyme involved here is what? Called as recombinase. PYQ alert. Recombinase enzyme is involved here. Recombination enzyme is also involved here. So what is happening? Recombinase enzyme. Recombinase enzyme also is involved here. Recombinase enzyme. Enzyme. The crossing over leads to what? Recombination of genetic material of the two chromosomes. So what is happening? Recombination is happening here. Recombination is happening. Recombination between homologous chromosome is completed by the end of Packeting. So end of Packeting, the crossing over has happened. Exchange of genetic material is over, leaving the chromosomes linked at the site of crossing over. They are linked at the site of crossing over. Any doubt in Packeting? Quickly ask me. Any doubt in Packeting? So what happened in Packeting basically? I'll summarize again. There are four main points something here. The first main point is that bivalent and tetrad are clearly visible in Packeting. Is that point clear to all of you? Is that point clear to all of you? In Pachydian stage, bivalent and tetrad are clearly visible. First point. Second point. Second point is that there is something called as crossing over. There's something called as crossing over is happening. What is crossing over? Exchange of genetic material between non-sister chromatids. Between non-sister chromatids of homologous chromosome. Crossing over. Second point is crossing over. Third point, recombinase enzyme. Recombinase enzyme. Fourth point is recombination nodule. Recombination nodules. From these 4 points, only the question will come in Pachydian. They will ask you, simple question, recombination nodules at which stage? You will tell me Pachydian. They'll ask you, bivalent is clearly visible in which state? You will tell me Pachydian. Crossing over happens in which stage? Pachydian again. Recombinase enzyme is present at which stage? Pachydian again. Recombination is happening which stage? Pachydian again. So do you realize how the question can be asked for Pachydian? How the question can be asked for Pachydian? Clear all of you? Any doubt here? Quickly ask me. Roshni, Tanuja, Krishna disappear, ran away somewhere. Yes? Any doubt in Pachydian? Any doubt in Pachydian, quickly students? No doubt? Awesome. Now, this is the crossing over. Can you see? Crossing over has taken place here. Crossing over has taken place. Here, crossing over has happened. Okay. Now, what is diplotin? What is diplotin, students? What is diplotin? Now here, what did we have in at the end of Pachydian? At the end of Pachydian, what did we have now, students? We have one chromosome here. Now we have one more chromosome. Okay. We have two chromosomes here. Oh, this chromosome looks a little weak. I don't know why. This is what we have. This is what we have at the end of Pachydian. At the end of Pachydian, this is what we have at the end of Pachydian. Now in diplotin, what happens, students? Diplotin, what happened? The first step in diplotin is what? The first step in diplotin is dissolving the entire synaptonemal complex. So what is happening, students? The first step is what? The first step is nothing but dissolution of synaptonemal complex. Everything is gone. Gone. The synaptonemal complex is gone. And the entire two chromosomes are free. The two chromosomes are free except at a one point. The two chromosomes are free now, except at one point. That one point here, can you see? This point right here, where it makes a X mark. Can you see? It makes a X mark here. And this point of attachment in which the only point where chromosomes are attached, that point right here is called as chiasmata. It is called as what? Chiasmata right here. Chiasmata. So the point of attachment at the only place where the two chromosomes are attached, that is called as chiasmata now. The X-shaped structure. What is it called? It's called as X-shaped structure. Shaped structure. Chiasmata. So what is the first step? First step is what? Dissolution of synaptonemal complex. Everything is dissolved completely. And the tendency of the recombined homologous chromosome of bivalent to separate from each other. See, homologous chromosomes are separating. Except at the site of crossing over. So whenever there is crossing over, in that place there nothing is happening. Okay. Now these X-shaped structures are called as chiasmata. Okay. Two main points of diplotin. But the third main point is very important. This also has been asked for PYQs because in oocyte, in the case of female also, human female, of some vertebrates, diplotin can last for months or years. This diplotin stage right here, a cell can be in diplotin stage for years together. Almost 10 years, a cell can be in diplotin stage. 10 years. A cell is stopped at diplotin, it is not going to the next stage only. 10 years or 12 years or 14 years, a cell can be stuck at diplotin stage. Three main points here. Three main points. The first one is what? The first one is dissolution of synaptonemal complex. The second one is chiasmata. Third one is arrest. Arrest. Okay? 3 points clear, all of you? There's 3 points clear to all of you. That is diplotin, 3 main points about diplotin. Okay. Now, the last step is called as diakinesis. The last step is called as diakinesis. What exactly is happening in diakinesis now? Now, students, see this. There was this X mark here. There was this X mark. Now, what exactly is happening is this X mark is also coming out. This X mark is also being completely dissolved. The X mark is being completely dissolved right now. That is called as dissolution of chiasmata or termination of chiasmata. The chiasmata, the X mark, is completely being removed. The chiasmata is being completely removed now. That is called as dissolution of chiasmata. Dissolution of chiasmata or termination of chiasmata. See here. This is marked by the termination of chiasmata. Gone. During this phase, the chromosomes are fully condensed again, because that is what happening in prophase, right? In prophase, the chromosomes are getting condensed. The mitotic apparent spindle is assembled to prepare the homologous chromosomes to separate. By the end of diakinesis, nucleolus disappear. Nuclear membrane also breaks down. Diakinesis represents the transition to the metaphase. Like how in the case of prophase of your mitosis, by the end of prophase, the chromosomes are getting condensed. By the end of late prophase, endoplasmic reticulum, everything was disappearing. In the case of meiosis also, by the end of diakinesis, end of prophase one, nucleolus, nuclear envelope, everything is disappearing. Everything is disappearing. Now, students, do you want a quick summary of everything? Tell me in the chat. Do you want a quick summary of entire prophase one? Can we do a quick summary? Main points I'll tell you again if you want. Tell me. Do you need a quick summary? Yes or no? Quickly in the chat. Tell me if you want a quick summary of everything. Before we go to the metaphase, let's do a quick summary of entire meiosis. Entire meiosis is a quick summary. Okay. The first phase of prophase. See, how many are there, students? First of all, tell me. There is leptotene, zygotene, pachytene, diplotin, and diakinesis. There are totally 5 stages. Totally 5 stages in prophase 1. Leptotene, zygotene, pachytene, diplotin, and diakinesis. 5 stages. What is happening in the first stage in leptotene, students? In leptotene, chromosomes are becoming gradually visible under light microscope, and condensation is starting. That's all. 2 main points. All of you. 2 main points about leptotene is that the stage is chromosomes are becoming gradually under light microscope, and the compaction is starting. Compaction of chromosomes is starting. 2 main points. Done. Leptotene done. Now, zygotene. The first main point about zygotene is synapses. What is synapses, students? Synapse is nothing but 2 chromosomes are coming together. That is called a synapses, pairing up. Now, between the 2 chromosomes, there is a gum or fevicol or feviquick. That gum or fevicol is called as synaptonemal complex. That is the second point here. First point is synapses. Second point is synaptonemal complex. Third point. Third point is that this stage is called as bivalent or tetrad. Formation is happening where? Formation is happening in zygotene. Formation of bivalent and tetrad is happening in zygotene. Why is it called as bivalent? Because of 2 chromosomes. Why is it called as tetrad? Because of 4 chromatids. Because of 4 different chromatids. Okay. Now, let's go to the next step. It is pachytene. What is happening in pachytene? There are 4 main points in pachytene. The first point is what? The bivalent or tetrad are more clearly visible now. Bivalent or tetrad are more clearly visible in the pachytene. Second point is the crossing over. Crossing over is happening. What is crossing over? Exchange of genetic material between non-sister chromatids. These are called as non-sister chromatids of homologous chromosomes. Second point, crossing over. Third point is recombination nodules. Formation of recombination nodules at the site of crossing over. Wherever crossing over is happening, recombination nodules formation happens there. The fourth point here. The fourth point is the enzyme mediated recombinase enzyme. 4 points pachytene. Now then we have the diplotin. Diplotin 2 main points, 3 main points diplotin again, 3 main points. First point is what? Dissolution of synaptonymal complex. The entire complex is being dissolved. Second point is what? Entire chromosomes are separated out except at one point. That is called as what? Chiasmata. That is called as chiasmata. That is also called as X-shape mark. X-shape mark. Third point is that the diploidy can last for many years. It can last for many years. Last is called as diakinesis. Main point is what? In diakinesis, termination of chiasmata. The chiasmata is terminating. First point. Second point is that the nuclear envelope, nucleolus, everything is disappearing. Condensation is completed of chromosomes. Condensation is completed of chromosomes. That is the entire Prophase I. Any doubt, quickly go, shoot me. Shoot your doubts, all of you. Any doubt in Prophase I? Any doubt in Prophase I? Your chat is visible. I'm looking at your chat. So, any doubt in Prophase I? No doubt? Then, now we go to metaphase now. Metaphase 2. So, metaphase 1. Now we go to metaphase 1. Now we go to metaphase 1. Now, before we understand metaphase 1, I want you to understand the mitosis metaphase first. Understand mitosis metaphase. In the case of mitosis, did we have something like this, students? In the case of mitosis metaphase, did we have something like this? Yes, we had something like this. Yes. The two chromosomes were separated. Now here we had the chromatid coming in, chromatid. We had the spindle fiber coming in, spindle fiber. Spindle fiber also was here. This was in mitosis. This was in mitosis. Now, students, in the case of, tell me, in the case of mitosis, did we have pairing of chromosomes? Tell me yes or no quickly in the chat. In the case of mitosis, did we have pairing of chromosomes? No. No pairing of chromosomes. But in the case of meiosis, students, in the case of meiosis, there is pairing of chromosomes. There is pairing of chromosomes. So can I tell, in the case of meiosis, here we have one chromosome. Here we have the other chromosome. Together what? Together they are the homologous chromosome. Homologous chromosome. Similarly, this is how it looks in meiosis. This is how it looks in meiosis. There are two equilateral plates. The two metaphasic plates are here, not one. There are two metaphasic plates here. Two metaphasic plates. Okay. Two metaphasic plates are here. And this is how the homologous chromosomes are attached. Okay. This is your bivalent. Bivalent. The bivalent chromosomes align on the equilateral plate. This is the equilateral plate. The microtubules from the opposite poles. Spindle fibers. Spindle fibers. Opposite poles of spindle fibers attached to kinetochore of homologous chromosome. Homologous chromosome. Now, students, can anybody tell me the difference between this and this now? With respect to spindle fiber and chromosome? What is the difference between mitosis and meiosis metaphase with respect to spindle fiber attachment to the kinetochore? Anyone in the chat who can tell me? What is the major difference? If you notice clearly, students, in the case of mitosis, one spindle fiber was attaching to one part of chromatid, one part of this kinetochore, same chromosome, one more chromatid, one more spindle fiber, right? For every chromosome had two spindle fiber attached to it. Can you see here also? One chromosome, two spindle fibers. But in the case of meiosis, do you see? One chromosome only has one chromatid now. So one spindle fiber. One chromosome has only one spindle fiber. One spindle fiber. But in the case of mitosis, one chromosome has two spindle fibers attaching. Clear? Is that point clear to all of you? Difference between mitosis and meiosis with respect to metaphase? Now we come to the anaphase. Understand? What exactly happened in the case of your mitosis, first of all? In the case of mitosis, in the case of mitosis, if this is the chromosome, if this is the chromosome, in the case of mitosis, the entire chromosome split into half. Entire centromere split into half, and we ended up getting two separate chromatids in the case of mitosis. The two chromatids were separated out. But in the case of meiosis, in the case of meiosis, that is not the case. In the case of meiosis, that is not the case. Because in the case of meiosis, what happening is, can you see here? If you go back, you can see here, there is no splitting of centromere here. No splitting of centromere. In fact, this chromosome is pulled back. This chromosome is being pulled back. This chromosome is pulled back. This entire chromosome is pulled back. Not a chromatid. The entire chromosomes are being pulled apart. That is, separation of homologous chromosome is happening in the case of anaphase. The homologous chromosomes separate while sister chromatids remain associated with the centromere. The sister chromatids will remain as such now. In the case of meiosis, sister chromatids will remain separate. There is separation of homologous chromosome. Now, if I ask you students, assertion in anaphase 1, in anaphase 1, centromere is splitting, true or false? Quickly go. In anaphase 1, centromere is splitting, true or false? Tell me. In the case of anaphase 1, centromere is splitting, true or false? All of you will tell me false, sir. False. Because centromere is not splitting in anaphase 1. There is separation of homologous chromosomes. False. There is only separation of homologous chromosomes. No splitting. No splitting is happening here. It's a false statement. False statement. Okay. That is anaphase. Can you see? The entire homologous chromosomes are being pulled apart. Entire chromosomes. The chromatid is intact. Chromatid is intact. Centromere is right there. Sister chromatids are as such. Okay, so chromatid as such. Now, now we come to telophase. Telophase is the opposite of prophase. All of you know. In telophase 1, the nuclear membrane and nucleolus are reappearing again. Cytokinesis follows this follows, and this is called as dyad of cells are formed. So after telophase, students, after telophase, you can see something called as dyad of cells. Dyad means two. You can see two cells after the telophase 1. Telophase 1. Clear? At the end of telophase 1, there are two different cells called as dyad of cells. Dyad of cells. Now, I might ask you, sir, what is the DNA content here? What is the DNA content here? And what is the chromosome number? So, I will tell you, students, I will tell you in telophase 1, this right here is nothing but N2C. This one is nothing but N2C. Now, why is it called as N2C? Stay till the very end to understand that. What is this N2C? Why there is only half the number of chromosomes and half the number of DNA content? N2C. Okay, N2C is here. That is N2C with two chromatids. With two chromatids. With two chromatids. Stay till the very end for that. Two chromatids. Okay. Although in many cases chromosomes do undergo some depression, they do not reach the extremely ends of the state of interphase nucleus. The stage between the two, meiotic division, is called as interkinase. I told you already in the starting. Between the two, meiosis 1 and meiosis 2, the between stage is called as what? Interkinase or interkinesis. Interkinesis. Okay. Interkinesis. Interkinesis is followed by prophase 2 as a much simpler prophase than prophase 1. So what do we undergo right now? We had Prophase I, Metaphase I, Anaphase I, and Telophase I. Now, what is happening here? Interkinase, interkinase, and then Prophase II. Prophase II is starting. Clear, all of you? Prophase II is starting after that. Now, can we start Prophase II, students? Now, tell me. Can we start Prophase II? Everyone in the chat. Can we start Prophase II? Yes. Very simple again. Very simple. I'll teach you one by line of NCERT. Understand every line of NCERT now. Okay. Prophase II. The meiosis II is initiated immediately after cytokinesis, usually before the chromosomes are fully elongated. In contrast to meiosis I, meiosis II resembles the normal mitosis. So, your meiosis II is just like mitosis. Mitosis. So, can I call meiosis II as equational division? Equational division, because it is just like mitosis. Equational division, yes, right. Nuclear, what happened to prophase? Nuclear membrane disappears by the end of prophase 2 and chromosome again becomes compact. That is compaction of chromosome happening. Compaction of chromosome. What happens in metaphase? All the chromosomes align at the equatorial plate. At the stage, chromosome align at the equatorial plate and spindle fiber attached to the kinetochore. What is happening in anaphase? Remember, students, in anaphase of mitosis, there was splitting of centromere. So, the splitting of centromere again in meiosis happens where? In the anaphase 2. So, anaphase 2, what happens? It is begins simultaneously by splitting the centromere. Centromere is split now. So, there is a famous question. There is a famous question now. Centromere splits in which two stages? In mitosis and meiosis. What should be your answer? Tell me. In mitosis and meiosis, in which stage centromere splits? Anaphase of mitosis and anaphase 2 of meiosis. I repeat again. Anaphase of mitosis and anaphase 2 of meiosis. In these two stages, the centromere is being split. Centromere is being split here. Clear? Of each chromosome, which was holding the sister chromosomes together, allowing them to move towards the opposite poles and shortening of microtubule and attached to kinetochore. Telophase again is opposite of prophase, again opposite. Meiosis II ends with telophase II, in which two group of chromosomes are once again gets enclosed by nuclear envelope is coming back. Envelope is coming back. Yes, welcome back. Cytokinesis is followed by resulting in the formation of tetrad, that is 4 haploid cells. 4 haploid cells are finally formed. So what we learned, students, right now? We learned that we have the first a diploid cell which undergoes what? Which undergoes something called as meiosis 1. In meiosis 1 we have 2 haploid cells. Now they undergo what? Now they undergo meiosis 2. They undergo meiosis 2, which makes again haploid cells. There is no change in ploidy. In meiosis, there is no change in ploidy. So 4 haploid cells at the end of meiosis. 4 haploid cells at the end of meiosis. 4 haploid daughter nuclei. Okay. Beautiful. Done. See, this is anaphase. Anaphase. Chromosomes are being pulled apart. This is telophase 2. Now, students, what is the DNA content here, students? The DNA content here is going to be NC. Here also is going to be NC. Now, what is this NC? I'll tell you again. I'll tell you now. NC has only one chromatid. NC has only one chromatid. One chromatid. That's all. One chromatid. Any doubt, students? Please ask me in the chat. Any doubt in the entire meiosis is over actually now. That is the end of meiosis. We started off with We started off with meiosis 1, we ended up meiosis 2. In the end, we got 4 haploid. In the end, we got 4 haploid daughter cells. Now I'll teach you something called as DNA content. Ready? Ready, students, for DNA content? Tell me in the chat. Ready for DNA content? If you're understanding, right? I believe all of you are understanding and following through. Any doubt anywhere, you can always ask me. It's very simple. I don't know why it's difficult for some students because they will try to mug it up. But when you understand with the help of diagrams, right, it becomes very easy. Okay. Can we start now, students? Mahima Yadav. Yes. Can we start? The DNA content. Understand each one of them. Any doubt, any point of DNA content also you can ask me in the chat. Okay. Now let's start with DNA content. Let me drink some water. All of you also drink some water. Today no break today because I'm already in the house. No requirement for break. I already ate my food and everything is over today. Now can we start DNA content? Understand, students. DNA content of human cell. Let's take a human cell. Let's take a human cell. How many chromosomes, students? In the case of human cell, how many chromosomes? 24 chromosomes. All of you know 24 chromosomes are there. 24 chromosomes. 24, huh? Or how much? Whatever it is, you believe, no? How many chromosomes? Tell me. Human cell. 46 chromosomes. I'm checking all of you. You're just randomly telling yes, yes, agreeing to everything. Don't agree to everything, okay? 46 chromosomes are there. And there's something called as DNA content. DNA content. DNA content is nothing but around, let's say, around 40 picograms. 40 picograms. 24 chromosomes. No, 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 no. There is 46 chromosomes in human cell. Diploid organisms, students, diploid, 2n. 23 pair of chromosomes, 23 pair. Don't believe whatever. Sometimes I'm testing all of you. Okay. I want to test all of you. Don't be very vigilant with me. Okay. Now let's 23 pair. Exactly. Right. Now we'll understand stages. We'll write stages here. Now we'll draw one line. Oh, I can't draw straight line. There's a problem here. Stage. Now here we have number of chromosomes. Number of chromosomes. Then we have something called as number of chromatids. Number of chromatids. Number of chromatids. And finally we have the DNA content. DNA content. Okay. Write on this much. First write on this much. Done. DNA content is here. Now let's start with the first one. Let's start with the first one. In G1 stage. In G1 stage. Number of chromosomes 46. Number of chromatid students. Number of chromatid students, tell me. 46 initially. DNA content is 40. Chromosomes look something like this. Like this, something like this. Now in S phase, what happens? The DNA content is doubling. This is becoming 92. This is becoming 80. So chromosomes will look something like this now. Okay. This is happening in S phase. S phase. Then we have something called as G2, Prophase, Metaphase. Everywhere, even in Anaphase, it is same to same. It is same to same. Everywhere it is same. Now, once we come to telophase, once we come to telophase, there are two things which you need to understand here. One is per cell, one is per nucleus. Nucleus. Per cell it is going to remain the same. Per cell it is going to remain the same. 46, 92, 80. 46, 92, 80. Remain the same. But per nucleus, let's go back here, friends. Let's go back to here. This is the telophase. This right here is telophase. Telophase. Right? Initially we had what? Initially we had 2n 2c. It became from 2n 2c it became 2n 4c. Yes, it became 2n 4c, right? Because DNA content double, number of chromosomes will not remain the same. Students, remember, in S phase, number of chromosomes will remain the same. DNA content is going to double. That's all. DNA content 2n 4c. So right now, your nucleus in the per cell is also at 2 and 4c. Per cell it is 2 and 4c only per cell. But oh, what happened? One second. Let me show something happened here. It came out. Okay. I hope it is visible now. I hope it is visible. This is Prophase I. Huh. It went back. Huh. This entire cell right now is how what? Entire cell right now is 2 and 4c. Right. The entire cell is right now 2 and 4c. Now students, the cell is dividing to 2. So can I tell out of this 2 and 4c, half is going this side, half is going this side. That is one number of one set of chromosomes going this side. Out of 46 chromosomes, 23 are going this side and 23 chromosomes are going this side. 23 chromosomes are going this side. Yes. Out of this 2 and 4c, 23 is going this side, 23 is going this side. Out of this 4c, 2c is going this side, 2c is going this side. Basically divide by 2. Divide by 2. Half is coming this side, half is coming this side. That is per nuclei. Per nuclei. So what is the DNA content now, students? So per cell is going to be, nucleus is going to be 23, half is 46, half is 40. This right now from 2 and 4 it is becoming N2C. N2C. Understood all of you? Tell me in the chat. N2C. Now what happens is we have Prophase II. We have Prophase II. Everything is going to remain the same. Metaphase II remain the same. Now again a change will come in Anaphase. Again a change will come in Anaphase right now. Why? Because in Anaphase, remember students, in Anaphase I told you, even in the case of mitosis, I told you each chromatid, even in the case of Anaphase, each chromatid will act like a chromosome. I told you. Did I tell you, students? Each chromatid will act like a chromosome. Tell me, how many chromatids do we have, students? Here, tell me, how many chromatids we have? Here we have right now 46 chromatids. 46 chromatids. Let me show you here. Let's go over here. Here we have how many? The same will come here, right? 46 here. So can I tell we have 46 chromatids now? Here, same thing is going to come down, right? So we have 46 chromatids. We have 46 chromatids right here. In the case of Anaphase II. In the case of Anaphase II, we have 46 chromatid. Now each chromatid is acting like a chromosome. So can I tell we have 46 chromosomes also? Yes. In Anaphase, it's very special. Always remember, temporarily in Anaphase, each chromatid is acting like a chromosome. Why? Because it's splitting up. Centromere is splitting up. Each chromatid here can become a chromosome for the daughter nuclei. Right? So each we have totally 46 chromatids and 46 chromosomes here. 46 chromosomes. Let's go back here. Anaphase, what do you have, students? Anaphase we have 46 chromosomes and 46 chromatids. DNA content is going to remain the same. Now finally we have the telophase. Telophase. Telophase again per cell per nuclei per nucleus. Per cell it is going to remain the same. Per cell is going to remain the same. Remain the same. But per nuclei it is going to change. Why? Let's see here. Now tell me students. Here we have how many cells totally? Entire telophase per cell. We have 46 chromosomes. 46 chromosomes we have, right? Can I tell from these 46 chromosomes, 23 are going this side, 23 are going this side. Yes or no? Tell me. Tell me, students. 23 is going this side and 23 is coming this side. Yes. Out of the 46 chromosomes in Anaphase. So here we have only N. But what happened to the DNA? DNA which was 40 DNA was there, 40 picogram was there. 20 is going this side and 20 is going this side. So what we have, students, here? We had here 2 and 2C. We had 2 and 2C temporarily here. 46 is 2 1, right? 46 is 2 1. This is 2C. This 2 and 2C is becoming what? Half of is what? Half is, this is 2 and 2C, right? So we have, let me erase that. 2 and 2C. Half is going this side again. Half is going this side again. Yes. So can I tell N, 1N is going this side. NC. 1C is coming this side. 1N is coming this side. 1C is coming this side. So NC. So by the end of telophase, we have 23 pair of chromosomes in each nuclei. Yes or no? So what do we have here? 23, 23 and just 20. Half it is becoming. Half it is becoming. It is getting converted to NC now. NC. That is my students. We started off with 2 and 2C. We start off with 2 and 2C and we're ending with NC. Can I call this as reduction division? Reduction division then. The entire thing. Entire meiosis is called as what? Entire meiosis is called as reduction division. There is a reduction in the number of chromosomes. Okay. Now you can ask me. Now you can ask me one small thing here. Sir, here you told me 20. 20 are here. So what is happening right now is we have here one diploid cell. How many picogram this was? 40 picogram. This was 40 picogram. It gave you two daughter cells, which was N, N. This gave two daughter cells later on after. That is, this is after meiosis 1. Meiosis 1. This is after meiosis 2. After meiosis 2. What do we have? N, 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 and N. Four of them. Now I'm telling you, this one is 20, this one is 20 picogram, this one is 20 picogram, this one also is 20 picogram. 20 plus 20, 40, 40, 60, 80. Here we have 80 picogram of DNA total. But in the starting we had 40 picogram. It doesn't make sense, no? What is happening here, students? What is happening here? Why doesn't it make sense? Why doesn't it make sense? Can anyone tell me? Can anyone tell me why it is not making sense? Because if you look back at this table right here, when the cell cycle started, in the S phase, didn't the duplication of DNA happen? 40 became 80 here. So can I tell students, can I tell this 40 became 80 in the S phase. Now this 80 is divided into 20, 20, 20 and 20 picogram of DNA. Beautiful. You cannot make mistake here. You cannot make a mistake here. That's how nature works. That is how nature works, students. Tell me, isn't it beautiful? Didn't you learn something very beautiful today? That is the beauty of meiosis. Beauty of meiosis. Now, some of you might be getting confused because if you're watching it for the first time, if you're learning meiosis like this for the first time, okay. So I would advise you to watch the lecture again at 2x speed and understand what I just taught to all of you here. And if you want to understand why I wrote here, how this anaphase this is working, for this anaphase to understand, you need to watch the first part where I've thought about mitosis also. Where I've thought about mitosis. Okay. So students, that is the end of today's class. Let's read the significance here. Last significance before we end today's class. Significance. Meiosis is the mechanism by which conservation of specific chromosome number of each species is achieved across generation in sexually reproducing organisms. That is, chromosome number is maintained. Chromosome number is maintained, right? From N2 to N to N. Again, we're getting N here. Now this N and this one more N will join together from egg. They'll end up becoming 2N again. The ploidy is maintained. Yes. Now, it also increases since crossing over is happening in meiosis. Since crossing over is happening, can I tell recombination is also happening? If recombination is happening, can I tell variations can come? If variations can come, then there is chance for evolution. So genetic variability in the population of organisms from one generation to the next, variations are very important for the process of evolution. So because of crossing over, variations are coming. Because of variation, evolution is happening. So if there was no meiosis, there would be no evolution at all. Okay? So it would be evolution would be there, but not at such high rate. Not at such a high rate. So that's it for today's class, students. And let me know in the comment section if you enjoyed meiosis, and quickly smash that like button and try sharing the session to all the friends who wants to understand meiosis in a depth and conceptual manner, and not just by heart it, not just by heart it. So thank you so much, students, for today's class, and I will see you all of you in the next class, all of you. Bye bye, take care.